Assalamu alaikum. In today's lecture, we'll be discussing about the morphological features of the gram-positive and gram-negative bacteria. We all know that bacteria is a typical prokaryotic cell and is microscopic in nature. We have to visualize this bacteria with the help of the microscope. We can use simple microscope to visualize this. For its visualization, we can introduce stain, which can be taken up by the bacterial cells, and these bacterial cells can be visualized under the simple compound microscope. We know that typical prokaryotes doesn't have membrane-bound organelle within in the cytoplasm. All the metabolic processes are taking place in the cytoplasm itself. It's either the replication process, transcription, translation, or other metabolic processes of the bacteria which are taking place for its growth and development are carried in the cytoplasm. Cytoplasm contain ribosomes, which are 70s. They contain genetic material in the nucleoid. They contain plasmid DNA, mesosomes, granules, and inclusion bodies, which includes starch, lipid, and sulfur. The nuclear region, where the genetic material is, this doesn't contain any nuclear envelope. This nucleoid is present in the cytoplasm itself. If we talk about the size of the size of the bacteria ranges from 1 micrometer to about 5 micrometer. If we talk about the shape of the bacteria, we are having different shapes of bacteria: cocci, bacillus, vibrio, spirilla, and spirochetes. Now the cocci. As you can see the diagram, cocci are spherical or oval cells. Bacillus are rod-shaped cells. Vibrios are comma-shaped curved rods. Comma-shaped curved rods. Spirilla are rigid spiral form, and spirochetes are flexuous spiral form. We are having one exception in case of bacteria, that is the mycoplasm. This mycoplasm is one of the bacteria which does not possess cell wall. Hence, they don't possess any stable morphology. They don't possess any stable morphology. We know that the shape of the bacteria is decided by the cell wall itself. Now, the structure of bacteria. If we talk about the simple structure of bacteria, starting from outside to inside, we are having glycocalyx. We are having glycocalyx, or we can say it's a outer structure is capsule or slime mold. Now, what is this glycocalyx? This glycocalyx is sticky, sugary envelope composed of polysaccharides and polypeptides. Polysaccharides and polypeptides. But this glycocalyx is firmly attached to the cell surface. जब क्या ये glycocalyx क्या है? Attach है cell wall के साथ, cell surface के साथ. That time we call this as a capsule. But when this glycocalyx doesn't have tough structure, it's water soluble and help cells to adhere to surface external of the cell. That time we call this as a slime layer. We are having flagella, we are having pli, we are having flagella, we are having pli, then move, then the cell wall, then the cytoplasmic wall, then the cytoplasm, then the cytoplasm. So we have to discuss about the cell wall. We have to discuss about the cell wall. The cell wall is used to give rigid shape to the bacteria, and it gives structural support to the bacterial cells. This cell wall protects bacterial cell from environment in extreme condition, whether it's deficit of nutrients, when there is extreme conditions like high temperature, high pH, or high salinity. If we talk about the bacterial cell wall, it's composed of peptidoglycans, which covers the entire cell surface of bacteria. These peptidoglycans, if we talk about the peptidoglycans, these peptidoglycans, as you can see in the diagram, these peptidoglycans are made up of a combination of peptide bond and carbohydrate. Peptide bond and carbohydrate. Either the carbohydrate will be N-acyl muramic acid or N-acyl glucosamine. N-acyl muramic acid. So that we represent by NAM, NAM, which is in green color here, which is shown in green color, and the N-acyl glucosamine, which is in orange color. So there is arrangement of NAG and NAM alternative in case of in peptidoglycans. On our screen, we are having typical cell wall structure for gram-positive bacteria. We are having typical structure for gram-positive bacteria. In case of gram-positive bacteria, we are having many layers of peptidoglycans. We are having many layers of peptidoglycan that retain the crystal of violet dye when we stain our bacterial cells. So when we use differential staining or when we use two different types of dyes, at that moment, this gram-positive bacteria is able to retain the primary color. And in case of differential staining, we are using primary stain, that is our crystal violet. So under microscope, we will be able to observe violet color rod or cocoa-shaped bacteria under the microscope. So starting from the base, we are having plasma membrane. This plasma membrane is made up of phospholipids, and in between the phospholipids, we are having integral membrane proteins or peripheral proteins are there. Then above, we are having the periplasmic space, which is in between the peptidoglycan layer and the plasma membrane. Then we are having the peptidoglycans. So in peptidoglycans, we are having alternation of N-acyl muramic acid and N-acyl glucosamine. N-acyl muramic acid and N-acyl glycosamine. So in case of gram-positive bacteria, the peptidoglycans, if we talk about now, particularly talk about the peptidoglycans, in case of gram-positive bacteria, the peptidoglycans is modified. It's heavily modified with carbohydrate bases. These carbohydrate bases are anionic polymer. That is, these carbohydrate bases are negatively charged polymer that play an important role in the membrane integrity. A major class of these cell surface glycopolymers are teichoic acid. These are actually the teichoic acid, which are phosphate rich molecules. So if they are phosphate rich molecules, it indicates that they are negatively charged. There are two types of teichoic acid: lipoteichoic acid, which are anchored to the plasma membrane, as you can see here, which are anchored to the plasma membrane and extend from the surface into the peptidoglycan. This teichoic acid is from peptidoglycan into the plasma membrane, means ये peptidoglycans को attach करता है plasma membrane के साथ, and that we call as lipoteichoic acid. That we call as lipoteichoic acid. And another is wall teichoic acid. Another is wall teichoic acid, which are covalently attached to peptidoglycan, which attach one peptidoglycan layer with the another one. This wall teichoic acid are covalently attached to peptidoglycan and extend through and beyond the cell wall, and extend through and beyond the cell wall. This was about the structure or feature of gram-positive bacteria. Now, what is major functions of this teichoic acid? Because its major component or one of the important parameters, one of the important components which is seen in the gram-positive bacteria, it gives negative charge. It gives negative charge. Its major role is in anchoring, transportation of ions, antigenic determinants, and external permeability barrier of bacteria. Now we will discuss about the cell wall of gram-positive bacteria. As you can see in the diagram, we are having typical structure for cell wall of gram-negative bacteria. The gram-negative bacteria have different cell envelope structure than the gram-positive bacteria. They have cytoplasmic membrane, or we call this as inner membrane or plasma membrane. Then we are having thin layer of peptidoglycans. In between peptidoglycans and plasma membrane, we are having space that is called as periplasmic surface. Then above we are having outer membrane. Then we are having outer membrane containing or made up of lipopolysaccharide. Lipopolysaccharide, and in this lipopolysaccharide, we are having proteins which act as a channel. We are having porin. We are having O-specific side chains. O-specific side chains for antigenic activity. Again, the plasma membrane is composed of phospholipids, and in between phospholipids, we are having integral membrane proteins. Then above, we are having the periplasmic space and 1 to 2 layer of peptidoglycans. This peptidoglycan is made up of NAM and NAG, and N-acetylmuramic acid and N-acetyl glucosamine. Again, this is connected with the outer membrane with the help of the teichoic acid. This lipopolysaccharide accounts for their endotoxic activity. We know that major of the disease is caused by gram-negative bacteria. It's because of the presence of the outer membrane above the peptidoglycan layer. This accounts for their endotoxic activity and antigen specificity. Lipopolysaccharides are consists of lipid A, core polysaccharides, and O antigen. And O antigen, which which is specific to gram-negative bacteria. We are having porins, and these porins act as a transporter of specific molecules from outside to inside. And most important, we are having bronze lipoproteins. This bronze lipoprotein act as a link between the outer membrane and the peptidoglycan chain by covalent bonding. So a bronze lipoproteins they connected this outer membrane with the peptidoglycans, as you can see the diagram. So this is the complete morphology of gram positive and gram negative bacteria.